Hi, thanks for joining me today. I'm Steve Williams, and I lead the capillary electrophoresis R&D group within the Genetic Sciences Division at Thermo Fisher Scientific. And today I'd like to highlight the role of Sanger sequencing and fragment analysis in the study of SARS-CoV-2. So by way of introduction and reminder, there are two different genetic analysis applications that run on the family of applied biosystems capillary electrophoresis systems. The first is Sanger sequencing using big diterminated chemistry, which is used to carry out standard DNA sequencing such as de novo sequencing, targeted resequencing, variant detection, and plasmid sequencing. And the second is fragment analysis, which uses fluorescently labeled primers to amplify specific regions of a target DNA to produce fragments of specific size. And this has a wide range of application, including quantitative fluorescence, PCR, microsatellite analysis, STR analysis for human identification forensic applications, and SNP genotyping. Thermo Fisher has a suite of CE genetic analyzers that are designed to meet the throughput needs of users. So we have a high throughput system, the 3730, which has 96 capillaries and integrated plate loader. We have a medium throughput system, the 3500 XL, which is either a 8 capillary system or a 24 capillary system, and also exists as a US IVD and CE IVD approved system. And then we have the latest addition to our instrument portfolio, which is the Seq Studio system, which is a benchtop instrument, very easy to use, and a 4 capillary system. And during today's presentation, I'll discuss how Sanger sequencing and fragment analysis can be used in the study of SARS-CoV-2. And I'll talk about various published articles as well as in-house work during the presentation, and give acknowledgement where appropriate via citation to the authors of those articles. So first, brief introduction to coronaviruses and the impact that they have had on human health. So I think, as everybody knows at this point in time, the coronaviruses are named for the crown-like spikes on their surface. And there are 4 main subgroups or genera of coronaviruses. These are alpha, beta, gamma, and delta. And coronaviruses, human coronaviruses, were first identified in the mid-1960s. And there are 7 coronaviruses that can infect humans. The first 4 in this list are the 229E, NL63, and OC43, and HKU1. And these all produce fairly mild symptoms, symptoms of a common cold in humans. And then there are 3 more serious viruses that produce much more severe respiratory infections. These are the Middle East Respiratory Syndrome, or MERS, and then the first version of SARS, and then the version of SARS that we're dealing with now, which is the virus that causes COVID-19. And these coronaviruses can infect animals and can evolve and then infect humans through this zoonosis process. So back in 2003, SARS, acute respiratory syndrome, SARS-CoV, the first version of SARS, was identified in China in 2002, 2003. It's killed, nearly 650 people in China and Hong Kong, and is thought to have been transmitted to humans via bats, and then into spits, and then on into humans. In 2012 was the syndromal MERS. This was identified in Saudi Arabia in 2012. And killed over 800 people in the Middle East, and was transferred from dromedary camels into humans. And now we're dealing with the latest strain of SARS-CoV-2, first identified at the end of 2019 in China, in Wuhan, China, and to date has infected approximately 30 million people around the world. I think we just passed the 30 million mark the other day, and we have close to 1 million deaths globally. and the virus is thought to be a market in Wuhan. And it's not currently known, the animal host for this virus, but it's thought to be bats again, possibly being transferred from bats to pangolins. But that remains to be determined, and the original source of the virus can take quite a while to determine. In the case of the first version of SARS, it took, I think, over a decade to verify the transmission route. So since the first whole genome of SARS-CoV-2 was published back in mid-January, scientists have sequenced tens of thousands of virus genomes. And this information has been really valuable for studying pathogen evolution and epidemic spread. So the next strain database is the main global repository for all of these sequences, and tens of thousands of sequences are now available. So NGS has, of course, been the primary technology used for whole genome sequencing, and there are many different published protocols on different NGS platforms that are available, including the IronSeq SARS-CoV-2 research panel, which provides 99% of the viral genome, and when it runs on the Genexus integrated sequencer, has a turnaround time of about 1 day to produce a full viral sequence. So for many of the NGS protocols, Sanger sequencing plays a role to help gap fill NGS sequences and to confirm identified variants. In fact, some of the very early sequencing data from groups in China use Sanger sequencing to gap fill the original NGS data. But in addition to that, there are 2 published protocols for complete genome sequencing of the SARS-CoV-2 virus. One from the CDC in the US and another from the CDC in China. So the first genome Sanger sequencing protocol was published by CDC back in March, shortly after the first sequence of the virus was published. The workflow is shown in the top of this slide. It begins with RNA extraction and then an RTPCR. In the case of samples that have so typically PCR CT values of 27 and above would require this second PCR step. PCR products are purified via exosapic purification and then a cycle sequence using the Big Determinator version 3.1 sequencing protocol, and then separated on the 3730 capillary electrophoresis instrument. And for the CDC protocol, there are 38 primer pairs that are needed to span the nearly 30,000 bases of the virus. And these are tiled across the virus, as shown in the top of the slide. And in the cases of a low viral load, where a second round nested PCR is performed, primers that sit just inside the original primers are used. This produces amplicons in the range of about 320 to 1,000 base pairs. And then 4 individual sequencing reactions are used to sequence both the forward and the reverse regions of each PCR product. So a total of 148 sequencing primers are used. And a single sample takes about 18 hours to sequence via this approach. And as I mentioned, this protocol is very useful for detecting low viral load samples. So the second published protocol shown in the bottom of the slide is a very similar approach. It tiles amplicons across the genome. In this case, also 38 primer pairs are used, producing amplicons in the range of 600 to 1,400 base pairs in length. Difference being here is that a rapid amplification of cDNA ends race approach is used to sequence the 3' and the 5' end of the virus. So there are a number of Sanger sequenced full genomes in the next strain database. Particularly from regions where access to expensive NGS instrumentation and expertise is limited. And also for samples that are very low viral load, Sanger sequencing is often the preferred choice. So beyond sequencing the entire genome, Sanger also has a significant role to help understand the host response to the virus. The life cycle of SARS-CoV-2 infection begins with the entry of the virus into the cell through the ACE2 receptor that's shown in the top left of the slide. And the spike region of the virus binds to ACE2 through the receptor binding domain of the virus. And variation in the ACE2 receptor sequence can affect how tightly the virus binds. It might alter a host susceptibility to SARS-CoV-2 infection and be responsible for person-to-person differences that are seen in infection. So studies have shown that there are about 120 amino acids on the outside of the outer surface of ACE2 that span the interface of the receptor binding domain of the virus. And there are two key hotspot mutations here in the alpha helical bundle. And these are important for SARS-CoV-2 binding. These are lysine 31 and lysine 353. And Sanger sequencing can be used to identify different variants in this region. So antibodies from vaccination or as a result of infection target the receptor binding domain of the spike protein. They neutralize the virus by binding in this region and block entry to the cell. So therapeutic neutralizing antibodies, also target this region of spike protein. And there's somewhere on the order of about 20 different monoclonal antibodies that are currently in clinical trials as treatment options for infection with coronavirus. And there are many more that are in preclinical development as well. So although mutation rates of the virus are slow, some changes in the receptor binding domain have been seen in some isolates. And it remains important to monitor the virus for changes of sequence in the receptor binding domain. So the domain is about 193 amino acids. So it can easily be sequenced by Sanger sequencing. And there's no need to sequence the entire virus for this purpose. So after the virus has entered the cell, newly formed virus is ingested by antigen-presenting cells, that's shown in the diagram, which go on to display fragments of the virus and can activate T helper cells, which then go on to turn on B cells, which produce antibodies against the virus, or to activate cytotoxic T cells, which can destroy the virus. So B cells can be isolated from patients infected with the virus by using the spike protein as bait, and then a technique like flow cytometry to isolate individual B cells. And there are well-established protocols for Sanger sequencing of heavy chain and light chain regions of antibodies that have been recovered from B cells. And so this is really useful for identifying tight binding antibody sequences. Finally, as we begin to think ahead to the prospect of an effective coronavirus vaccine next year, the question of durability of response in an individual is an important question. And there's a growing body of research that links immune response to variations in the microbiome composition present in our bodies. So there are as many as 100 times more bacterial genes residing in humans than human genes. And recent data indicates the gut microbiota can modulate the response to vaccines. And as we'll see in the next few slides, a fragment analysis-based CE approach can provide a rapid, cost-effective approach to profiling microbiota in the gut and the airways. So a technique known as Isospro has been developed by a small startup company in the Netherlands called InBiome. And their approach is based on the specific amplification of interspace regions in bacteria to generate fluorescently labeled fragments that can then be separated on Applied Biosystems genetic analyzer instruments. So all bacteria have at least one interspace region, and many bacteria have multiple regions. And Isospro relies on length and sequence polymorphisms of these interspace regions to provide identification of the bacteria. So, for example, in the bottom of this slide show these interspace regions for this particular bacterial species, Enterococcus faecalis. it has 4 different interspace regions: IS1, IS2, IS3, and IS4. After amplification of IS1 and IS2, you get a 377 base pair fragment, and amplification of IS3 and IS4 produces 275 base pair fragment. And so each bacteria has an almost unique fingerprint when these interspace regions are amplified. And you can also group different phylum of bacteria by using different labeled primers. So 3 different colors shown here for 3 different phyla. And some of the advantages of this approach are that it's a rapid workflow. You can get an answer within about a 4 to 5 hour turnaround time. Does not require culture of the bacteria, and it can detect unknown and unknown species. No need to predefine the species that you're looking for. And in addition, it can detect bacteria that cannot be cultivated. So our colleagues in Biome have used this approach to examine microbiota in nasopharyngeal swabs collected from patients, individuals that have been infected with a SARS-CoV-2. And they have used this technique to identify specific microbiota profiles that may be correlated to the likelihood of infection of the virus. They examined 135 different clinical research samples from people ranging in age from the very young to the very elderly in this 0 to 100 age range. And they used the Isospro assay to profile DNA isolated from these nasopharyngeal swabs. So what you can see on the slide is a resulting clusterogram with a PCR status of each individual is listed either as red or green, red being positive for the virus, green being negative for the virus. And these were independently determined by qPCR. And then you can see that, after unsupervised clustering was performed of all of the profiles, there's a clear profile of the 77 samples with similar microbiota profile in the center of the clusterogram here. And in addition, so this cluster of this central cluster showed a very low level or a relatively low level of SARS-CoV-2 positivity compared to the cluster that was outside of the central cluster. So for the cluster, about 25% of the people were SARS-CoV-2 positive, and outside of this cluster, this rose to 47%. And in addition, data that's not shown in this slide indicates that older individuals were actually enriched for this H signature, which poses the question whether there is an age-dependent pharyngeal microbiota signature that has an increased susceptibility. So further and more detailed analysis of this data set can be found in this publication that's listed on the slide, or alternatively, there is a poster that will also be presented in this conference that goes into more detail into the data analysis and the conclusions. So I want to switch gears now and talk a little bit about the use of Sanger zoonotic. So there's wide recognition of zoonotic disease as a growing threat. In fact, the CDC estimates that more than 6 out of every 10 people can be spread from animals. And in fact, 3 out of 4 new or emerging infectious diseases actually pass animals into people. And we can pass either directly from an animal into a human. For example, as we discussed, dromedaries were responsible for directly transferring the MERS virus. And of course, we have bird flu and swine flu that can be directly transferred into humans, or they can start from a viral source such as a bat and pass through another animal, either a civet, as in the case of the original SARS, or it's postulated that the pangolins are the intermediary hosts for the SARS-CoV-2 virus. So there's a growing need for assays that can detect pathogens that can pass from humans to animals to humans, and also to identify the natural reservoir for a particular pathogen that has the possibility of infecting the human population. So sequencing can be used to address this, to provide information on pathogens in animal hosts that are closely related to humanized versions and more likely to cross the species boundary. So one approach that has been used for this purpose has capability to identify all currently recognized and potentially future emergent coronaviruses is an RT-PCR amplification-based approach, followed by sequencing of a particular region of the coronavirus, this RNA-dependent RNA polymerase region of the virus in the ORF1AB region. And this approach is described in the publication listed at the top of the slide here in the Journal of Clinical Virology, but has been widely used by others as well. The approach consists of two different PCRs that use degenerate primers. A first PCR that produces an amplicon about 670 bases in length with degenerate forward and reverse primers, and then a second PCR that is a nested approach that generates a smaller fragment, again using a degenerate primer of about 600 base pairs. And this amplicon can then be Sanger sequenced to confirm the identity. So with this approach, there are a large number of coronaviruses that can be identified, and the assay shows virtually no cross-reactivity with other common respiratory pathogens such as RSV or flu. And it can be used to routinely screen individuals whose occupation bring them into close contact with animals for prolonged periods of time. Or it can be used for routine surveillance in species such as bats, which are reservoirs for viruses with human pathogenic potential. So finally, I'd like to briefly describe an application of CE in the study of SARS-CoV-2 detection that is based on a fragment analysis approach. The workflow for CE fragment analysis detection is actually similar to qPCR. It starts with a collection of the sample via nasopharyngeal swab, and then extraction of the RNA, for example, using one of the kits from Thermo Fisher, a Magmax viral pathogen nucleic acid isolation kit. And then after RNA extraction, an RT-PCR step is performed with fluorescently labeled primers. And at the end of this, the fragments, fluorescently labeled fragments that have been amplified are identified on the CE. And the size of the fragment and the color of the fragment encodes data about which amplicon is being detected. And the data can be analyzed via a software such as GeneMapper from Thermo Fisher. So for the assay developed at Thermo Fisher, there were 3 different regions of the virus that were amplified. One from the ORF1AB region, the second from the spike gene, and the third from the nucleocapsid gene. And this results in 3 different size fragments. An N protein fragment at 396 base pairs, an ORF1AB at 425, and S protein at 437 base pairs. And additionally, an RNA positive control is added to the reaction to help check for the presence of any PCR inhibitors that might prevent amplification. So one added feature of this assay that I'd like to point out is the use of multiple fluorescently labeled primers. This allows, after amplification, for the samples to be combined, to be pooled together, such that 4 individual reactions can be run on the capillary, combined and read out and run on the CE. And the fragments are then identified based on not only the size, but also the color. So each capillary can then produce answers for the application from 4 individuals. And the sensitivity of this approach is similar to qPCR methods, and more details on the protocol and an application note can be found on the Thermo Fisher website as shown on the slide, including the sequences of all of the primers that are used in the assay. So by way of summary, hopefully what I've shown you is that Sanger sequencing and fragment analysis technologies are widely useful in the detection and sequencing of SARS-CoV-2. Sanger sequencing can be used to support NGS, whole genome sequencing by gap filling and confirmation of variants, but can also be used to generate whole genome sequence by itself. Targeted sequencing, can be used to look at SARS-CoV-2 host response factors, such as the sequence of ACE2 receptors, which the virus binds to gain entry to the cell, or can be used to look at the immune response, look at the antibody sequences that are produced from individual B cells by sequencing of the variable regions. There has been an assay developed that amplifies and sequences a highly conserved region of the virus, the RDPRP region, and this can be useful to identify the natural reservoir of the virus or to help track animal coronaviruses that may jump to species. And then finally, bacterial profiling using the Isospro technology can develop a more comprehensive understanding of how the microbiome can influence host response to pathogen infection. So with that, I'll conclude my talk and invite any questions, and thank you for your time.